The overall goal of this procedure is to use double thymidine synchronization and high-resolution confocal microscopy to study the mitotic roles of multifunctional proteins that may possess critical interphase functions.
This method can help answer key questions in the cell biology field about how to delineate the novel functions of proteins involved in cell cycle and mitosis. The main advantages of this technique are that the cells can maintain their normal physiological behavior and that this method is also very easy to perform. Demonstrating this procedure will be Dr. Mohammed Amin, a postdoc from my laboratory, who is an expert in the use of this method.
For fixed cell imaging of mitotic cell progression, begin by seeding approximately 2 × 10⁵ HeLa cells into each well of a 6-well plate containing a 70% ethanol and UV sterilized cover slip and 2 ml of DMEM medium. After 24 hours in a cell culture incubator, block the cells with 2 ml of freshly diluted thymidine in fresh medium per well, and return the plate to the incubator for another 18 hours. The next day, wash the cells 2 times with 2 ml of PBS and 1 time with fresh 37°C medium. Return the cells to the incubator for 9 hours to release the cells from the block, followed by the addition of another 2 ml of thymidine supplemented medium per well. After the second blocking incubation, wash the cells 2 times with 2 ml of PBS and 1 time with 2 ml of fresh 37°C medium, and add 100 nanomolar of freshly prepared